Amino acids, they undergo several reactions. The first reaction is whenever I have two cysteine molecules that are next to each other. Cysteine is the only amino acid that has a thiol, which is an SH functional group. So if we have two cysteine molecules that are next to each other, and cysteine is a neutral amino acid that has a polar side chain. So whenever these two cysteine molecules are next to each other, one cysteine will lose a hydrogen, the other one will lose an SH, and what I will form is the disulfide bridge. It's the SS bridge. This is one of the characteristics and one of the main reactions cysteine undergoes whenever two amino acid cysteine units, they are next to each other. Another reaction, this one explains how two amino acids connect with each other. So one end of the amino acid, which is the carboxylic, will lose an OH, and the other end of the amino acid will lose a hydrogen. So this means that I'm losing water, and I'm going to connect together, and I'm going to form an amide linkage, which I'm going to call it a peptide. It's a peptide bond. This is a peptide bond, and this bond connects two amino acids with each other. So one amino acid will lose an OH, the other one will lose the hydrogen from the amine, and you are going to connect those two amino acids by a peptide linkage or an amide linkage. Both are the same. Okay, so if you connect two amino acids together, this is going to be a dipeptide. And if you connect the three amino acids together, it's going to be a tripeptide. Tetrapeptide is whenever you have four amino acids connected together, and so forth. So pentapeptide is 5 amino acids, and as you go. Now, how do you connect amino acids together? The order is very important because two different dipeptides can be formed whenever two unique amino acids react. So let's have an example. Let's say if I want to connect glycine and alanine. So all what you need to do is just go to the table, write the structure of glycine, write the structure of alanine, but always make sure that the amino group is to the left of every single amino acid, okay? And the reaction simply goes as such. Because this is a base and this is an acid, the base normally takes the acidic hydrogen, so that becomes an OH. And then the acid loses the OH and the amino loses a hydrogen. You connect those together by a peptide or an amide linkage, and this is how you are forming the dipeptide. The sequence is important because if you react glycine and alanine, then you're going to get a dipeptide which starts with glycine and ends with alanine. In order to break down that dipeptide to its individual amino acids, all what you need to do is just to break down the amide bond. And this is the glycylalanine dipeptide. I can change the sequence, and instead of starting with the glycine, I can start with alanine. So if I start with alanine and glycine, then I'm going to end up getting the alanyl glycine dipeptide. This is completely different than that. Different properties of the dipeptides. So it's completely different with and what amino acid you want to form. So peptides, these are amino acids. These are of short chains. They can be dipeptide, tri, tetra, penta. These are short chains. You're just connecting those amino acid units together. Now, when you have more than 10 amino acids, up to 50 amino acids, then we're going to call this a polypeptide. So a polypeptide is an intermediate chain length polymer with less than 50 amino acids. Once we talk about proteins, these are polymers. And these polymers, they have more than 50 amino acids. So we want to differentiate between polypeptides and proteins. Proteins, they have a lot of amino acids in them, more than 50. So 50 and more, that is called a protein. 50 and less, up to 10 carbons, this is called a polypeptide. And then depending less than 10 amino acids, depending on how many amino acids you have, you can call it di, tri, tetra, penta, hexa, hepta, and so forth, all the way to deca peptides. Amino acid residue, this is an amino acid that is part of the peptide, polypeptide, or the protein chain. Whenever you want to write down the peptide, always peptides, they are written with the N-terminal residue on the left. Okay, so here I do have a sequence of tripeptide. So this one is a tripeptide. How did I know that it's a tripeptide? I knew that it is a tripeptide in there because I do have 3 amino acids that are connected together. So the first one, I can simply break it down. So the first one over here, break down the amide bond. Look at the table, and the first one here, this is valine. Okay. Then you can break down the second one. Always break down the amide bond in there. And that, again, look at the table. This is going to be glycine. This is the simplest amino acid. So that is going to be glycine in there. And then the last amino acid here, so this whole thing with the CH3, okay? So that whole thing is with the CH3. So the last amino acid here, let's write it like that. So that last amino acid over here, that is going to be leucine. So here I do have a peptide chain, which is made up of 3 amino acids. In order to connect those together, I always need to start from the N-terminal of one amino acid, and I should end up with the C-terminal of the other of the last amino acid. Here my connection is valine, glycine, and leucine. Okay, this is how I write it down. It's valine, glycine, and leucine. So I should end with the C terminal. This means that the carboxylate of the leucine. And I should start with the N terminal. This means that the amino of the valine. Let's have some problems. These problems, they do have a solution. I'm just going to walk you through these. So write the 2 reactions to represent the formation of 2 dipeptides that form when valine and serine, they react with each other. So react with each other. So all what you need to do is just go to the table and check the structure for valine and serine. Now, they did not indicate the sequence of the dipeptide that I need to form. So I can do valine, serine, or serine, valine. Okay, so I'm going to end up getting 2 dipeptides as the question saying in there. So over here, what I have is the valine that is reacting with the serine in there. So I'm going to always start with the N-terminal of the first amino acid in there. So that's the N-terminal of the amino acid. So you always do NC. This is it. So the N is always to the left, and this is going to be the C in there. When I do a reaction and I am making the peptide bond, the carboxylate will lose the O, or you can do a reaction between the O and the hydrogen in there, make this an OH, and the OH is lost and the hydrogen is lost from the other side, whatever you want. So here I am making the peptide bond. I'm going to lose an oxygen there, and I'm going to lose one of the hydrogen, actually 2 of the hydrogens here to make water. So I will end up forming an NH over here that is directly connected to the carbonyl, and that is going to be my amide bond that I am forming to connect those 2 amino acids, the valine and the serine in there. So if you want to go backwards and you want to break the bond, then all what you need to do is simply go ahead and break the amide bond in there so that you can get the individual amino acids. You add an oxygen here and you add two hydrogens in there, and you get your two amino acids that you started with. I can start with the serine and the valine. Always make sure that you are starting with the N terminal. And when you write your dipeptide or your peptide chain, make sure that you start with the N terminal, you end up with the C terminal, which is the carboxylate in there. So over here, I'm going to lose the oxygen, I'm going to lose two hydrogens, I'm going to combine everything together and form my amide bond. Another problem, write the complete structural formula and the abbreviated formula. They need a tripeptide, and that tripeptide needs to be formed from aspartate, cysteine, and valine. Now, I need to indicate to you the sequence of the connectivity of these three amino acids in the question. So here it says that in which the C terminal is cysteine. The C terminal is cysteine. This means that you end in cysteine, okay? And the N terminal is valine. This means that you have to start with valine. So go ahead and check your amino acid table in here and write the structures of valine. Definitely, we will have over here the aspartate in the middle. So you have your valine. You have your aspartate. So here you go. That is the aspartate. And that is going to be the cysteine in there. So go ahead and write the structures for each one of them and allow the valine to lose an oxygen, the aspartate to lose two hydrogens, and so forth, and connect those amino acids together. Okay, in this problem here, they want it's the same thing, and they gave you that the C terminal residue is the serine and the N terminal residue is the phenylalanine. So I need to start with the phenylalanine and I need to end up with the serine, and then I can connect all of these amino acids together. This is the answer in there. So as you did before, go to the table of the amino acids, write down the structures of each of these amino acids. And once you write down the structures of these amino acids in there, you can allow the water to leave and you can form the peptide bond. Whenever I ask you about the different isomers, check the connectivity. So here I'm working with a tripeptide. Tripeptide means that I have three amino acids that are connected together. And always with tripeptides, the number of isomers is going to be 6, meaning that I would have 6 different combinations. So write the abbreviated formula for the 6 isomeric tripeptides of alanine, phenylalanine, and arginine. So just connect them together because we do have different possibilities in there. Alanine can be either at the beginning, it can be in the middle, for example, over here, and it can be at the end. Question 5 is the same. For 6, how many tripeptide isomers that contain 1 glycine, 2 alanine? So this is going to make your tripeptide. So go ahead and do the combination, and we're going to end up getting 3 tripeptides that satisfy this condition.